Hi class, in this video we're going to talk about some details of protein synthesis. We know that in protein synthesis that we are taking the code that is found in DNA, so it's a sequence of DNA bases that are found in a molecule of DNA, and this segment of DNA bases has the code, or like the blueprint or the recipe for making a protein. And that sequence of bases in DNA is called the gene. So we're going to talk specifically about how we go from having just the recipe, the DNA code, the gene, to making protein. We have two steps we're going to go through. One is called transcription and the other is called translation. In these notes, we're going to talk about the details and what happens in transcription and translation. So first of all, the overview, the main part of the atom. This is a eukaryotic cell. We know it's a eukaryotic cell because it has a nucleus and has organized organelles in it. In a eukaryotic organelle, like this one, this is an animal cell, the genetic material, the DNA, is found inside of the nucleus. So we have chromosomes that are here inside the nucleus, and we want to get the information from those chromosomes in order to make protein. Now, say we want to make a protein to grow some hair, for example. So maybe the gene that contains the instructions for growing the hair is just in there between the two brackets. So the first thing we have to do is we need to make a copy of just this target gene that's in between these two brackets right here. So I'm going to change colors, and I'm going to make a copy of just those things that are here in the brackets. And that copy is something called messenger RNA, and that process is called transcription. So I've made a copy of just the recipe that I need. So think of all the chromosomes as being a recipe book that contains all the recipes. Well, if you only want to make one certain recipe, like just say make Rice Krispie treats, you only need to have that recipe. So in this example here, we are taking making a recipe or a copy of just the recipe that we need, and that is called transcription. Then this piece of messenger RNA leaves the nucleus, and it goes out and it docks on a ribosome. When it's here at a ribosome, there's another molecule, which I'll do like this blue color, another molecule that brings structures called amino acids. Amino acids are basically the things that make a protein. And messenger RNA tells it what it needs. So messenger RNA's got the recipe, and it's going to tell these little blue molecules that I drew right here called tRNA to bring in the amino acids, which I have here in brown. So here's the amino acids here, in order to basically make a chain of amino acids here. This chain of amino acids is the protein. So messenger RNA has the instructions that it got from the nucleus. It brings it out to the ribosome, and then transfer RNA brings it the amino acids that it needs to actually construct the protein. So like Rice Krispie treats in this example. Once it has made its protein, then the protein moves over to this structure right here, and this is the rough endoplasmic reticulum. So the protein goes inside the rough endoplasmic reticulum where it gets processed. Once it is done processing, it goes in a little bubble called a vesicle, and it goes over here to the Golgi apparatus, which then kind of packages it. And so over here it gets modified in the rough endoplasmic reticulum, and then it goes to the Golgi where it gets like the packaging. And then it decides what to do with the protein. The Golgi decides if it's going to keep the protein for something it needs, or it's going to export it. A lot of proteins are exported. It just depends what the protein is. So what we're going to be talking about in these notes today is how do we make messenger RNA? How do we transcribe? How do we get that recipe from the chromosome? And how do we end up using that recipe to make the actual protein? So transcription is here, the first part, when we make messenger RNA. And translation is here, where we use the recipe we got from DNA in order to make the actual protein. So the first part of the notes, I'm going to talk about just transcription. Again, transcription is just this part here, where we are making a piece of messenger RNA from the DNA template. So here is DNA. We can tell this is DNA for two reasons. First of all, it's double-stranded, so you can see one strand right here and the other strand right here. And we can see that it's got T's in it. I know it's kind of blurry, not the best, but it's got T's that are right here. So what we're going to need to do is we're going to need to uncoil the DNA. Now the DNA is long and thin, and we may only need this region. That's where the gene is that has the instructions for our protein. So only that region has to uncoil. and it uncoils 10 to 20 nucleotides at a time. Now, a nucleotide is a base, a sugar, and a phosphate. So this whole thing here is a nucleotide. So it's going to uncoil basically one base at a time, until 10 to 20 segments are open. So it's going to continue to open. So eventually when we see this gene, it's going to look like this. It's going to be like flat here, and then it's going to be open like a loop here in the middle. So it's going to uncoil just where it needs to uncoil, just at that target gene, which is, the gene that has the instructions. Now, it's going to be pried apart by an enzyme. An enzyme is just a protein molecule that makes a chemical reaction occur. In this case right here, the enzyme, what it's doing is it's breaking the bonds that held them together. So you can see here, here's like, say this is a T and this is an A, held together by a bond here. What the enzyme does is it breaks those bonds so that the molecule can be opened up. And this right here, from here to here, this is the target gene. So this is the gene that has the sequence to make the protein. Now there's going to be two different codes. So there's going to be a code up here that maybe this is T, A, and C, and then down here this would have been A, T, and G. So there's two different codes depending if you read the top or the bottom. So we're going to be looking for the template strand. The template strand. Now there's a way of figuring it out, but you don't have to worry about that for my class. I'm going to tell you which strand is the template strand. So one strand gets read, the other strand doesn't. It's kind of like a holding place for it. So our next step is now we got our enzyme that has opened up our DNA molecule. So now we are ready to start building our copy, a piece of messenger RNA. Before we get to that, I have a quiz question. So we know the entire DNA strand is really long. Each DNA strand, each chromosome can contain thousands of genes, depending on the length of the chromosome. So some chromosomes are longer than others. So some are really long, some are much shorter. But either way, they all contain many different genes. So does it make sense to open up the entire strand, or does it make sense just to open up the target gene like that right there? So does the entire DNA strand uncoil during transcription? No, it does not. It doesn't make sense because the cell will be wasting a lot of energy opening the whole thing if all it needs is just that one area right there. So does it uncoil during transcription, the entire thing? No, just at the target gene that it needs. Now that we've got it open our target gene, you can see that it's closed up here and closed down here and only open here at our target gene right here. So now that it's open off our target gene, now we need to make our mRNA piece off the DNA template. Now in DNA, we have 4 different bases. So in DNA, we have adenine pairs with thymine every time, like puzzle pieces together, and cytosine pairs with guanine. Now RNA, there is no such thing as T or thymine in RNA. In RNA, A pairs with uracil and cytosine pairs with guanine. Now this can be a little bit difficult when you're talking about base pairing rules. Now if you're looking at this picture here, over here this is like the placeholder strand, and this is the one that's the template. We can tell it's the template because messenger RNA, which is here in green here, is pairing off of it. So notice here that A goes with U, A goes with U, but the T that was here in DNA does go with A. So if we're making kind of a chart, if the DNA code versus the RNA code, and this type of RNA is called messenger RNA. So if it's T in DNA, it's going to be A in mRNA. If it's A in DNA, it's going to be U. If it's C, it's going to be G. And if it's G, it's going to be C. So C and G always go together, but the A's and the T's get a little goofy. So we're basically making our piece of messenger RNA here from the DNA template. So the first letter is U, U, A, U, U, C, G, G, U, and so on. So our DNA is opened up. We're making our piece of messenger RNA now. When it gets to the end of the gene, then basically the mRNA strand that was made right here is going to disconnect from the DNA template and float away. It's literally going to float away and leave the nucleus. The DNA is open, you can see here, at the target gene, but it won't be like that for long. It's like magnets. They will snap back together as soon as they can. Now, before mRNA leaves the nucleus, it goes through processing. Notice this only happens in eukaryotic cells. And eukaryotic cells are cells with a nucleus. So like plant and animal cells, insects, things like that. Things like bacteria, this is not the case. Those are prokaryotic cells. So in eukaryotic cells, we have some processing that happens. And so like a cap and a tail is put on. So the cap here, you can see in blue, a tail is put on right here. The reason for a cap and a tail is to protect the mRNA from getting like broken down by enzymes by accident. And it will also help it attach to the ribosome later. And also some parts of the RNA molecule are cut out. So some things are, you can see here, the green parts were cut out and the red parts were put together. So they call that splicing. So RNA is processed a little bit after it disconnects from DNA, and then now it's going to leave the nucleus. After mRNA leaves DNA, like I said, it completely recoils and goes back to normal. DNA is done. Remember, DNA is like the big recipe book that has all of the recipes in it. But the only recipe it gave for us in our example is the recipe for Rice Krispie Treats. So now messenger RNA has the recipe for Rice Krispie Treats, and that's it. So mRNA will now leave, and it's going to go out, and it's going to dock on the closest ribosome, the first one that it runs into right here. So it's going to leave. So RNA is made here. It's going to leave. It's going to come out and dock on the closest ribosome. That is the end of transcription. I have an animation I want to show you, and I'm only going to show you a little bit of it, and we're going to come back to it a little bit later. So this first part we're going to look at, and we'll come back to it later. So just watch this for right now. Amoeba Sisters.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information it codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's a big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips.
We're just going to pause here for right now, and we'll come back to that. Let me see if I can find my notes. There I am. So we're going to come back to that. So in transcription, we'll just stop pausing there for right now. We'll come back to the video. Transcription is the first process where we have our DNA template and we end up making our mRNA strand that then will go to the ribosome and go through translation. So we'll come back to that video. So looking here, I'm going to change my color to blue. So this is just some practice. So practice transcribing this piece of DNA. How do we know this is DNA? Because it's got T's in it. So if there's T's in it, then that means that we're looking at DNA. Now remember, there's two strands of DNA. So how do you know which one's the template strand? Well, this one I've already given it to you. Let's go over base pairing rules before we get started. So if DNA says one thing, what would mRNA say? So if DNA says T, mRNA would be A. If DNA says A, mRNA would be U. And DNA is C, it'd be G, and G, and C. So if we're trying to figure out and make our piece of mRNA, here's our DNA right here, but we want to make mRNA, we're going to be transcribing that. So in this case, T goes with A. A goes with U. I'm just following the base pairing rules that we just wrote on the bottom. C goes with G. G goes with C. So I'm just following the base pairing rules. A goes with U, C goes with G, G goes with C, T goes with A, C goes with G, C, U, C, U, U, C, C, G, A, U, G, C. So that's our transcription. So this is our mRNA transcript that's here. For the next slide, and you see it on your note sheet too, and you're going to be filling out your note sheet as you're going through these notes. I want you to pause the video and take a moment and write it out for yourself, and then check your answers to see if you're right. So I'm going to do the second strand here. So again, this is pieces DNA. It's right here. We wrote the mRNA and DNA base pairing rules right there on the slide previous. So I'm going to make my mRNA transcript following my rules. So T goes with A. So hopefully you've done this already. Uh-oh. It does this sometimes when I touch it in a funny way. So A, U. So hopefully you have already written it out yourself and you're just checking your answers. And if you got something wrong, you just fix it. And I find my students come with a lot of different backgrounds. Some people have learned this stuff before and some people haven't. So some of you it might be easy, some of you it might be a little bit harder. So that is what your mRNA transcript should be. That's right there. So again, after mRNA is made, it now gets processed, it leaves the nucleus, and it goes up to the ribosome for the second step. The second step is called translation. So basically, transcription, remember, occurs here where you have your piece of DNA. You just make a copy of your target gene, which I'll do in, whoops, maybe I won't. Oh, maybe I won't again. Let me try, maybe that worked. All right, there it is. So I just made a piece of mRNA here, and that is called transcription. So what is produced in transcription? Messenger RNA. That's what's produced in transcription. Where does transcription occur? It occurs in the nucleus. At the end of transcription, messenger RNA leaves, and it goes out and it docks on the closest ribosome. And this is now where translation starts. So in translation, we're going to basically be building the protein. So I'm going to show you this overview video next. Just pretty short. Make sure I have the right video here. And just watch it.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so that video went over both transcription and translation. And the last one did not. It just went over transcription so far. So we're going to kind of pick up from where we left off. So we know that we have transcription. Our transcript has come. So we know we have our messenger RNA that has arrived here at the ribosome. And that's the end of transcription. Now that it's arrived, now we're going to move to the next part. And we're going to be having to deal with something called amino acids. Amino acids are the building blocks for proteins. So like, for example, those of you who may work out, you may have amino acid shakes or protein bars that people eat before they work out because they supply them with amino acids. So amino acids are the building blocks of proteins. There are 20 different types of amino acids. You don't need to remember that, but there are 20 different types. And the number of amino acids determines what kind of protein it is, the order of amino acids, and the arrangement of the amino acids. So just like letters in a word. So like, for example, if you had 3 letters in a word, if you had different letters, then that would change the amino acid, would change the word, change the protein. If you had them in a different order, that would change the protein as well. Or if you had a different number of letters, a different number of amino acids, that would change it. So amino acids are like the letters of the word, and the word would be like the protein that's there. Another way to think of it is we're using Rice Krispie treats as an example. So Rice Krispie treats has several ingredients like Rice Krispies, butter, marshmallows. Maybe you put chocolate chips in your Rice Krispie treats. Those are all things, those ingredients are like amino acids. So when you combine different ingredients, like if you put hot sauce in your Rice Krispie treats, then that would change that, but be like adding a different amino acid. So amino acids will be built into the whole protein. So the ingredients would be the amino acids in our example, and Rice Krispie treats would be the protein in our example. So we've got, at the end of this step right here, we have messenger RNA that is now docked here on the ribosome in the cytoplasm of the cell. Now we're going to be dealing with something called transfer RNA, which looks funky. Transfer RNA looks like this. On one end, it has an amino acid. What amino acid it has, because like I said, there are 20, depends on the code that's down here. So, for example, all tRNAs that have the code UGG all carry the same exact amino acid. It will come and it'll bring the amino acid to the ribosome, drop it off, and then go pick up another amino acid in case it needs to be used again. So transfer has this unique shape. So the actual steps of translation. So we've got our piece of messenger RNA that is docked there on the ribosome. The ribosome is going to bind, and in the video they showed that the first amino acid, the first transfer RNA came and bound. The first tRNA always, 100% of the time, brings an amino acid called methionine. Methionine, they call it the start, so the start of it here. So the very first amino acid that's brought here is methionine. Now here is the mRNA code that we already transcribed for DNA. Now notice the base pairing rules here are a little bit different. So on our mRNA code versus tRNA code. Now because they're both RNA, they do not have thymine, they do not have T's, they have U's, uracil. So if mRNA has an A, then tRNA will be U. If mRNA has a U, it would have an A, and C, and G, and G, and C. No such thing as a T here. So here is mRNA right here, the bottom one. This whole strand here is mRNA. So notice that A here goes with U goes with A, and G goes with C. These 3 bases on mRNA are called a codon. And these 3 bases on tRNA are called the anticodon, so it matches with them. So what amino acid it brings depends on the codons of the mRNA and the tRNA. So the very first one, 100% of the time, is methionine. And then what comes next depends on what the next code is. And we have a chart, we can figure it out. So you don't ever have to memorize what code comes next. There's an easy way of figuring it out. So the tRNA brings the next one, and it's going to attach it to methionine. And then this one's going to come in next here, this lysine, and it's going to attach it to that one there. So it's going to eventually make like a bunch of poppet beads of amino acids that are together. So each next thing that comes in will bring another amino acid. So let's just pretend that the next bases are C, G, and A. And that means the anticodon would be G, C, and U, and it would have an amino acid that's attached to it that's going to drop off. So which amino acid comes next depends on the code in messenger RNA. So you're going to end up seeing a molecule like this. Like I said, it looks like a bunch of poppet beads that are together. Now, what kind of a protein that this is making right here depends on the types of poppet beads, so the types of ingredients, the number of ingredients, and the order of the ingredients. Those things all matter. So it's going to eventually make this big string of poppet beads of ingredients until it reaches the end of the piece of messenger RNA. Once it reaches the end, then the protein, all these poppet beads, will disconnect from the mRNA, and it'll disconnect from the ribosome to eventually coil up and become a protein. It's called a polypeptide until it coils up. So now I want to look at the second animation. So it's the first animation that we saw. We're just going to see the second half. And I'm going to rewind it just a tad because I went a little too far when I played it the first time. So I want to rewind it just a few seconds. Let's start here.
It's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in, and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA. CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So, the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so just know that the process of transcription and translation can be difficult, and we're talking about it now. Sometimes watching the video again, this entire video can be helpful. Sometimes watching the Amoeba Sisters video again can be helpful. But we're also going to be covering it in lab and stuff as well too. So hopefully after a while it'll kind of all stick. But realize it can be a complicated process. Here's kind of the last quiz questions with the main ideas, the big things that I want you to get out of this. First of all, where does transcription occur? So transcription is the first process. It's the first thing that happens, and transcription occurs. I'm trying to go to my picture. It's closer. Here in the nucleus. So transcription here occurs in the nucleus. So where does transcription occur for the first quiz question? Sorry if I'm making you dizzy there. It's the nucleus. What do we make in transcription? What's produced in transcription? We make a piece of messenger RNA. Low power. Okay, I better hurry. Messenger RNA. And then translation is the second half. Remember that mRNA leaves the nucleus and it goes out to the ribosome. So where does translation occur? You could say translation occurs in the ribosome. And ribosomes are found in the cytoplasm of the cell. So really either answer is correct. What is produced in translation? The protein. The protein is what's produced there in that. And that's the end of the notes. So hopefully you've completed your template. And then notice that there's a summary. So write a few sentences about the overall summary of the notes in that last section and then turn it in. All right, thank you. Talk to you soon.